Hi everyone. In this video, I'm going to be going over the embryology of the brain. So this is going to be a three-part video lecture series in substitution for chapter 6 of brain facts. Okay, so in this three-part video series, I'm going to be going over these materials. So first, what is embryology? And then in this video, I'm going to go over the stages from zygote to embryo. And then after that, in the second video, I'm going to go over the neural plate, crest cells, and neural tube formation. And then in the last video, I'm going to go over some anomalies in development. Okay, so first, what is embryology? So neuroembryology is the branch of embryology that deals with the origin and development of the nervous system. So in simple terms, it basically explores how, when you're a baby, how your nervous system forms. Um, why is this important? So first, it gives us clues for functions of structures. It gives us an understanding of normal development. And if you understand normal development, you can understand the causes and signs of abnormal development, as well as congenital anomalies. Congenital meaning by birth, anomalies meaning abnormalities. Neuroembryology is also heavily used in evolution to see how structures have evolved from species to species. So first, let's go into the precursor steps. So just as a preface, this is not tested in the brain beat. However, these materials are really helpful to understanding the preface for neuron development and the brain development. So first, you have on day 5. So let's start from the basics, right? You have an egg, your oocyte or your ovum, and your sperm cell, which will merge together to form a zygote, a fertilized embryo, right? So basically what happens is that embryo, that zygote, will differentiate into the inner cell mass and your outer cell mass, right? So your inner cell mass will, on day 9, differentiate into the bilaminar disc. Bi meaning two, laminar meaning layer. So the bilaminar disc means the two-layer disc. So you have your epiblast cells and your hypoblast cells. So this big rectangle doesn't actually represent the disc itself. It represents the cavity. So what's the actual disc is just a segment of that. So it would be just this portion over here. Only the dark portion would be the bilaminar disc. This outer portion would be the amniotic cavity. So your epiblast cells line the amniotic cavity, and then your hypoblast cells also line the yolk sac, and part of those are part of the bilaminar disc, if that makes sense. Okay, so now that we get an orientation of this, the most important step occurs on day 12, which is known as gastrulation. So here is a zoomed in version of the bilaminar disc, with the purple cells being your epiblast and your yellow cells being your hypoblast. Remember, epiblast is usually your amnion and your hypoblast is your yolk sac. And the way I remember this is that your epiblast cells, epi, the root epi translates to outer. So here, let's try writing some things down to get a better understanding. I'm back. Basically, oops, sorry, the annotate menu is being a little weird right now. But okay. So when you say epiblast, right? Epi translates to outer. Just like, so basically, you know how the amniotic fluid surrounds the embryo? It like cushions it. It's on the outside of the embryo, which is why it's the amniotic cavity. So the amnion. And hypoblast, hypo means inner or like under, and it's under the amniotic cavity, so it's the yolk sac. But I like to use epi to mean amnion, and then I just remember hypoblast as not amnion, which means yolk sac. Okay, so the reason day 12 is so important is it shifts that bilaminar disc, which is known as a two-layer disc, into a trilaminar disc. And Before I get into all the nitpicky details, I'll give you a general gist of what happens. So the cells on the outside will start moving inwards and will push the hypoblast cells. So the epiblast cells will start moving inwards and will push away these cells to start creating a new layer, a new inner layer, which would be your middle layer. Let's change the color to help you get a better understanding. So what it's going to do is the epiblast cells move inwards and will push away the hypoblast cells and will form your mesodermal layer or your middle layer, right? So let's get more into the specifics of how this happens. So I'm going to use this really helpful picture over here. So this is first your cell mass, right? And what's going to happen is on your posterior end, you're going to have a small bump forming, and this is called your primitive streak, okay? And this bundle of cells will start moving from posterior to anterior. So from your anal end, your butt end, to your head end, so your cranial end, right? And as it keeps moving in, cells will start moving inward. So your epiblast cells will start moving inwards towards the little groove that's forming your primitive streak. The cells are going to start moving inward. And towards some point, there's going to be a small structure called the primitive node forming. So that marks the end of the primitive streak, and it's formed mostly towards the cranial end, right? And what this does is it releases special molecules called signaling molecules, which basically transform the epiblast cells into this middle layer type of cells, okay? So this is going to make a little bit more sense when we go on to the second slide, but what you need to understand is that it's going to transform epiblast cells into a different type of cells, which is not epiblast nor hypoblast, but some middle type of cells, right? And then finally, after all of this, you can see here in this diagram how the cells, the epiblast cells, once they're pushed inwards, become kind of a different color. They're not blue anymore. They're now red, but they're also not yellow, which means they're a completely different type of cells, right? So now what you're going to officially have is three different layers of cells. Your endoderm, which are purely hypoblast cells, your mesoderm, which is that intermediate type of cells, and your ectodermal cells. So your ectodermal cells are your topmost layer. So let's move on to the next slide. Okay, sorry. So now remember we used to have a two-layer disc. Now what we have is something known as our trilaminar disc. Remember, tri translates to three and laminar translates to layer, so a three-layer disc. So why that second type, why I was emphasizing that this is an intermediate type of cell, is that all these three layers have different types of stem cells almost. So undifferentiated cells. Basically, let's go over the definition of differentiated because you're going to be hearing this term a lot now. Differentiated. And this actually is not specific to neurology. This happens all over the body. Okay, so what differentiated means, right, is every single cell type originates from kind of like a template cell. So this cell is known as your stem cells, your progenitor cells. They have a variety of name. These stem cells can take on any function, right? They are not assigned a function yet. So what happens in the middle is that certain molecules known as signaling molecules will come in and will change these stem cells. So basically, this is a complex process. Basically, the signaling molecule will come and bind to receptors in or on the cell and will go into the nucleus and cause the expression of certain genes. That's a little beyond the scope of the Brain Facts and Brain Beat book, but I will make a separate video on this. It's known as a process of gene regulation and gene expression. It's actually a really interesting mechanism. And basically what ends up happening is that by expressing certain genes, you can produce proteins which give these stem cells their own distinctive shape and function. That's really important, okay? So they give them their distinctive shape and their distinctive function. So an example of a specialized cell would be your neurons, actually. Your neurons, they have their own shape. They have kind of like a multipolar. So I'm going to take multipolar neurons as an example, which are basically neurons that have multiple branches. So see here, this is the nucleus, this is the cell body, these are the dendrites, the axon, and then your little axon terminals. So notice how it has its own distinctive shape, and it also has its own distinctive function of receiving, processing, and transmitting information. Another example would be your skeletal muscles, right? So, or your smooth muscles, better said, because these are a little bit easier to draw. They're thin, spindle shaped, and have centrally located nuclei. And the reason this is so important is because this cell type, smooth muscles, are usually used for constant contraction. So this is usually found in your digestive system, where in your intestines where they're constantly pushing food along your intestinal walls, right? So this and the shape is useful because it can endure long periods of pressure, constant pressure, right? So this has its own distinctive shape and its distinctive function. Like look at how different these two cells look, right? Um, and the reason all of this is important is basically both of these cells originated from a base type of cell, from this cell, right? And these stem cells, what happens is originally when you're a baby, your inner cell mass is made up of one type of stem cell. And then do you remember when we talked about the formation of the bilaminar disc? Then it becomes two different types of stem cells. And then now, after we have your trilaminar disc formation, every layer has a different type of stem cell, which is really important, right? Because they have different functions altogether. So basically, these three layers are the basis of all the organs that'll be formed. So let's go more deep into that. First, let's start off with your endoderm. Your endoderm is this thin yellow lining over here. And basically what it is it's going to form your inner lining of your gut tube. So basically when you're talking about your intestines, your stomach, everything, we refer to it as one continuous tube, right? If you think about it, that is what it is. And the reason we can call it this is because the intestines, your bowels, they all have the same internal four layers, except there are some minor altercations in the tubes. And so we call it the gut tube. It forms the inner lining of the gut tube. It also will form the inner lining of your lungs. So basically, when you have your lungs, the innermost layer is known as your mucosa, and your submucosa, all of that is derived from your endoderm. And basically, when you think endoderm, just think inner lining of organs. Your mesoderm forms your cardiovascular system, your muscles, your bones. There's a few more details in this section over here if you wanted to take a look at that. And the reason that's so important is that these mesodermal stem cells have the precursors to form the heart and the bones. But if you were to put ectodermal cells, they could not form heart and bones, right? Or if you were to take endodermal cells and ask them to form the heart, they couldn't. And likewise, if you ask the mesodermal cells or you try to make the mesodermal cells form the lining of the digestive tract, they couldn't because they don't have the capacity to do that. They don't have the specific regulatory factors, genes to do that, right? And then finally, the most important layer for our lecture series is the ectoderm. This is something that you will definitely have to memorize. The ectodermal layer is where the brain is derived from. It forms the central nervous system, and it forms a lot of the epidermis of the skin, and it forms basically your nerve, your brain, and your spinal cord. That's really important, and that was actually a question that I had at my regional brain bee, and I highly suggest you memorize that. Okay. And that's it for this video. In the next video, we're going to be going over the actual formation of the neural tube, the neural plate, neural crest cells, so on and so forth. And that's actually one of my favorite units. And just as a brief disclaimer, embryology is really hard. Like that was the unit that I struggled with the most. So I am going to try to make this as easy as possible to kind of understand. And I've kind of put together a timeline that and a compilation of all the important materials in this lecture series. But at the same time, I will acknowledge that my video series is not as extensive as I know some people would want them to be. So I will also put a list of videos that I recommend that animate this whole process so that it's easier to understand, because looking at it in 2D is completely different than looking at it in 3D and seeing how everything forms and molds. So on the last video of the series, I'm going to put together a bunch of resources and I'll publish that out. So yeah, let me know if you have any suggestions for me. And that I also promised you guys a bank of resources. I am working on that and it should be posted by the end of this month. So by June 31st, there should be a link on my bio that has all my resources. I'm just trying to get them reviewed by professionals to make sure that there aren't any anomalies or any like incorrect information. Thank you guys for your continual patience. Bye.